Hello, thank you to be at the end of this intense and very interesting day about molecular ecology. Well, this is not my talk exactly because it should be the talk of Irene Villalta, my last postdoc, that she did all the genetic analysis of this paper. And it was also in collaboration with Raphael Boulet. I want to this is a tribute also to Raphael Boulet, who passed away 1 year ago. And we have been working during around 20 years together. And also, this work was the master thesis of Leah Rami, our student of master thesis. Well, the first thing is to explain a little bit about ants. What are the ants? What are cuticular hydrocarbons? Well, I think that everyone knows very well what is an ant, but the role of cuticular hydrocarbons, cuticular lipids, is the layer under the cuticle are lipids, is a role about the chemical communication is used for nestmate recognition from the colony recognition to know if it's an intruder or they belong to our own colony and they have. But in the origin of these cuticular lipids, it was a waterproofing system. These lipids are to prevent the desiccation, and the main role is also for water balance. And this is a trade-off with chemical communication and water balance because there are different kinds of cuticular hydrocarbons, and the most important cuticular hydrocarbons that protect ants against desiccation are the saturated and linear hydrocarbons. And the two questions that we try to answer here in this work is if these cuticular hydrocarbons vary along a temperature gradient and if they are genetically determined. Our study system is this antispecies, Phenogaster iberica. It's a very common species in southern Spain. It's an endemic species, Thermophilus. Colonies are relatively small with around 1,000 workers with only one queen, and this queen only mates with one male. They are monogynous and they are monoandrous. And our study system was in Sierra Nevada mountains here. Well, I think that you know very well where is Sierra Nevada, in an altitudinal gradient from 100 to 2,000 meters of altitude. This is our 8 different populations that we have been working. And well, as you can see, more or less all the populations are very similar. It's open habitats where these species live all the times in shrublands or with scarce isolated trees. For the method, well, we studied first the cuticular hydrocarbons with a lot of colonies per population in these 8 populations, and we did a quantitative cuticular hydrocarbons analysis, analyzing the peak areas from the chromatograms. For genetic analysis were more or less the same, only one worker per colony for a lot of colonies per populations in these 8 populations. And the sequencing was done by a genus screen in France with 8 microsatellites and with the PCR. And the next major recognition by assays were dyadic encounters, putting one worker facing another worker. The controls were the same colony, and the tests were with all the combinations of colonies that belonged to the same population or to other population. We did an aggression score, was no aggression, and one it was aggression. In the case of aggression, this is an example. You can see how this worker is biting the leg of the other worker, is biting only, sometimes it's not necessarily biting, it's only mandible opening, or they are pulling appendages. For the results of cuticular hydrocarbons, we found in total 14 in alkanes, the linear alkanes, and also methyl branched alkanes, and the most abundant were changed carbons of 27 carbons or 29 carbons, and we found significant difference among populations. The first result was that we found how when the proportion of the different kinds of alkanes is different according with the altitudinal gradient. Any alkanes, the linear alkanes, decrease the proportion when altitude increase. Why? Well, because these alkanes are expensive to produce for the ant, and at the same time, in these high altitude populations, they don't need these alkanes to protect against the heat, because these populations are cooler, and they don't need this enormous quantity of N-alkanes, and they have only methyl branched alkanes. Even when we analyze the effect of altitude, but taking into account the size of the ant, we find the same trend. We can find that we can see how the quantity of N-alkanes decrease with elevation. Concerning population genetics, we found 38 haplotypes, and each haplotype was found in a single population. The greatest, as you can see by the colors, more or less, and the greatest haplotype diversity was at intermediate elevations. It's 600 and 1,000 meters of altitude. Concerning the network of haplotypes, we have found that neighbor populations have more similar haplotypes with the population of 1,000 meters. That was the exception, because they had two related haplotypes related to 301 haplogroup that it was related with 300 and 600 meters populations, and another haplogroup related to 300 and 700 meters of altitude. But there are no signs of local spatial segregation in these haplogroups in the population of 1,000 meters. We found also a high level of genetic differentiation except in the neighboring mid-elevation populations. We can see how all but 7 in red pairwise comparisons out of the 28 comparisons between of the populations were significant. And isolation by distance was related to the elevation, but not to the horizontal distance. Using the genetic distance, we can see how the genetic distance increase with the elevation, but there are no relationships, according with the mantle test, with the genetic distance and the horizontal distance. Aggression also increased with elevation and also with the horizontal distances. And aggression increases also with genetic distance and with chemical dissimilarity, with cuticular hydrocarbon dissimilarity. This correlation between aggression and cuticular hydrocarbon dissimilarity remained significant even after accounting for the correlation between genetic distance and aggression. In conclusion, we can conclude that as elevation increase, temperature in these high altitude populations, temperature decrease, and in this case, ants don't need large amounts of N-alkanes that are expensive to produce. And that also these populations at higher elevations are genetically isolated, and this situation could promote local adaptations or other phenomena, but we have not deep in this subject. And concerning cuticular hydrocarbons, this similarity is correlated with the aggression, but the changes within populations of these cuticular hydrocarbons have not effect on aggression. Environmental and genetic constraints may affect cuticular hydrocarbons profiles, but not among competing colonies within the same population. Okay, thank you for your attention, and also I want to thank Cristela Sanchez-Homs, that was our last PhD student, that she was working also in the same altitudinal gradient, and Charlotte Hemingway, that helped us for field work. Thank you.